and I'll show you later how to make a custom study deck. And let's go over this and see how long it takes us for 20 flashcards. So hemoglobin is a blank structure protein characteristically made of 2 alpha and 2 beta subunits. So it's a, what kind of structure protein? It's a globular quaternary structure protein. We got it wrong, so we definitely are going for a gain. You may see the other units introduced in your passage that you do not have to memorize coming in. For example, fetal HbF is made of 2 alpha and 2 gamma subunits, while sickle cell anemia is a disease due to a lack of functional beta subunit, and thalassemias are underproduction of units leading to weird HB like HBH or HB warts. So this is a very good slide. So we need to know that hemoglobins are a perfect example for quaternary structures. The subunits are globules, right? Globins, not globule. Wait a second. So now we need to ask, is globulin different from globin? Yes, globulin and globin, I meant. Globulin or globin, I meant. Globulin refers to a group of proteins found in the blood plasma. These proteins include various types, such as alpha, beta, and gamma globulins. They serve different functions, including immune system support, antibodies, blood clotting, and transportation of different substances in blood. Globin, on the other hand, specifically refers to the protein component found in hemoglobin, the molecule within red blood cells responsible for carrying oxygen. So globin is different, completely different. It's specific to hemoglobins, and it is composed of globin chains, either alpha or beta globin chains, depending on the type, each bound to a heme group that holds an iron atom, enabling oxygen transport. So it is worth it to find a picture of hemoglobin or globin per se. This is a perfect example. So we're going to put it here. Perfect. Okay. What is a non-protein molecule that binds to a protein and is needed to help that protein carry out its function? So it should be a coenzyme, but it's a cofactor. So non-protein. Did we write here to see what are the differences between? No. So is globin different from globulin? Yes. The next question that we are dealing with right now is what is the difference between cofactor and coenzyme? While ChatGPT is answering this, I remember here that we had cofactors here. So cofactors are metal cations that is required by some enzymes, but coenzymes are organic, like protein, carbohydrate, fat, molecules that is required by some enzymes. So, interestingly, hemoglobin is cofactor. A cofactor is a non-protein molecule. That's the first sign. It's inorganic. It's like a metal cation. And HB's hemoglobin's main cofactor is called heme, and it's this factor that contains the iron characteristically associated with blood. So let's also find a picture for heme. So this is a good one. Let's put it here. Perfect. Close. You can see how this whole structure, I think there's aromaticity. Is there aromaticity in the heme? Let's see what ChatGPT answered. Cofactors are non-protein chemical compounds or ions that assist enzymes in catalyzing reactions. They can be divided into two types: inorganic cofactors and organic cofactors, which are coenzymes. Inorganic cofactors can be metal ions like zinc, iron, magnesium, or copper, which help enzymes in their catalytic activity. It can also be small molecules or ions that bind to the enzyme's active site to enable enzymatic function. Metal ions like Mg2+, zinc 2+, or ferritin 3+ are inorganic cofactors. For instance, zinc ions in enzymes like carbonic anhydrase are essential for their catalytic activity, and cofactors aid. Their function is basically facilitating the proper folding of the enzyme, stabilizing the enzyme substrate complex, or participating directly in the catalytic reaction. And coenzymes are organic. Examples are NADH, NADPH, ATP, CoA, among others. For instance, NADH and NADPH act as carriers of electrons in redox reactions. And their functions are carrying and transferring specific atoms. So what are cofactor and coenzyme examples? So here we can add coenzyme examples, and also cofactor examples. It was the zinc for carbonic anhydrase. Cofactors are inorganic. Coenzymes are organic because they are coenzyme. It's easier because they're the enzyme. Okay. Cool. What describes a protein's graduated response to substrate binding? So, what describes a protein's graduated response to substrate binding? It would be cooperativity, meaning the addition or subtraction of a substrate can cause the affinity of a protein to that substrate to increase or decrease. So cooperativity describes the protein's graduated response to substrate binding. We have positive cooperativity for hemoglobin, right? What does it mean when it's said that a protein has positive cooperativity? Positive cooperativity refers to phenomena observed in proteins, especially in enzymes or proteins involved in binding other molecules, where the binding of one molecule to protein increases the affinity or ability of subsequent molecules to bind to other sites on the protein. So, can you give me examples other than hemoglobin? So in hemoglobin, the protein responsible for oxygen transport in red blood cells, when the oxygen binds to one subunit of hemoglobin, it alters the protein's conformation, making it easier for subsequent oxygen molecules to bind to the remaining subunits. So potassium channels also display positive cooperativity. As ions bind to one side, it can influence the probability or ease of opening additional ion channels. I think we see that in the neurons, right? So enzymes with multiple subunits and the enzyme aspartate transcarbamylase ATCase is involved in pyrimidine biosynthesis. Pyrimidine biosynthesis. So we had purines and pyrimidines. Purines were Ag and pyrimidines were Cg, right? We have for muscle contraction troponin and tropomyosin. They're in the sarcomeres of muscle fibers. They exhibit positive cooperativity. Calcium ions binding to troponin facilitates the binding of additional calcium ions, leading to muscle contraction. And for allosteric enzymes, for instance, PFK1, a key regulatory enzyme in glycolysis, exhibits positive cooperativity with its F6P. Some DNA binding proteins, transcription factors, can display positive cooperativity in binding. Once one subunit binds to DNA, it may facilitate the binding of other subunits and receptors. Ligand-gated receptors, nicotinic acetylcholine receptors, can demonstrate positive cooperativity in ligand binding. The binding of one ligand enhances the affinity for additional ligands. Perfect examples. Amazing. Okay, I have to go, and we'll be back in a few hours, in a couple hours, and continue with the rest of this. So for now, this is amazing. Yes, we have it here. One of them is due, and amazing. Ciao for now.